I'd like to begin by thanking Dennis Lowe and his colleagues for inviting me to give this talk, but I don't want to thank him or them at all for giving me this task. Nobody knows what the future anti-cancer targets would be. And so for that reason, my talk is actually named after Ojibwe Indian phrase called Ni Miakana, which means my path of thinking. So 350 years ago, French playwright said, doctors pour drugs, of which they know little, to cure disease, of which they know less, into patients of whom they know nothing. That actually remained for another 200 years until about the 1900s, where of course cancer was then treated by surgery, radiation, and drugs. Now you notice there's Goodman and Gilman were the first to really administer drugs for cancer. Does anybody in the audience actually know what that drug was? Since I don't have a lot of time, I'm going to move on. In 1942-43, the Germans bombed Bari, Italy, in the hope that they could win the war. But it was too late. The Americans and the British warships were in the harbor. Within 10 days, there were 2 waves of deaths. The first wave of civilian and military deaths were due to the bombs. The second wave was unexplained. Stuart Alexander, American medical officer, said, "What's going on?" But he noticed that the lymph nodes of these patients were smaller. And the smell of garlic in the air, he concluded it was nitrogen mustard. Meanwhile, at Yale University, Goodman and Gilman was working on lymphomas, where patients have cancer of the lymph nodes. The 2 events gave us what is now known as chemotherapy. And the first one was indeed nitrogen mustard. No new chemotherapeutic agents are now being made by major pharmaceutical companies. And it's time to move on. Then came the discovery of oncogenes in 1976 by Stalin, Varmus, Bishop, and Vogt. Oncogenes, in essence, are genes that have a gain of function either by amplification of the genes or a gain of mutation of the gene, allowing the gene to function faster and out of sequence. Targeting oncogenes is like targeting the horses, because if you shoot the horses, you stop the cart. That went on for 25 years with great success. The first one being Herceptin, and the most recent one is the targeting of a mutation in KRAS. Since, as you heard, that hundreds of thousands of patients have been sequenced, there are no new major oncogenes. It's time to move on. How do we move on? You can't target the horses, because there are no new ones, you have to target the cards. What are the cards? The cards are the transformed states of the cancer cell as a consequence of the action of oncogenes and tumor suppressor genes. And what are the cards? The cards are metabolic, aneuploidy, and immune. So what are these things doing? They are there to upset the cell. Our cells is like what Aesop said, like it is passion. It's like fire and water. They're good servants, but they're bad masters, because biology and physiology has, through millions of years, came to an understanding that there is a cohesion. There is a whole symphony. So how do you target metabolism when there are 2,500 genes? Genetics, genetics, genetics. Our laboratory's been doing that for the last 20 years. One of the mutations that came upon, and was first discovered by Bert Augustine at Johns Hopkins University, and our laboratory discovered the same mutation in T-cell lymphoma with the French lab of Philippe Goulart. Isocitrate dehydrogenase. It cannot be more metabolic, can it? These mutations cause gliomas, T-cell lymphomas, leukemias, etc., etc. So how does it work? Two postdocs, Japanese postdoc and a German postdoc, then made the first knock-in mice. We made a mouse as if the mouse had leukemia due to this metabolic enzyme mutation. And in so doing, together with the company Agios that I helped co-founded, we discovered this is not a gain of function, it's not a loss of function. The mutations create an oncometabolite. This metabolite is not known to exist, but it causes cancer when it's there. More recently, we discovered very interesting tumor microenvironment play. The mutation in the T cells bring the B cells in. So in normal lymph node, where you make antibodies, the T cells and the B cells interact to make better antibodies. In this case, the T cells and the B cells party and party and party beyond what it is supposed to do. But at the same time, the cells do not die by downregulating the death signal fast. What's the consequence? The consequence is T-cell lymphoma. Even more so, that even though the mutations are on the T cells, the B cells who come in with the partying occasionally develop B-cell lymphoma that we call shaking hands with the devil. Together with Craig Thompson and Luke Cantley, we started the company Agios. We developed drugs against these metabolic enzymes, and the first two were approved for acute myeloblastic leukemia back in 2017 and 2018. The third drug very recently obtained a very encouraging note. Our collaborator, Xavier, a French pharmaceutical company, ran a phase 3 trial on deadly glioma, and it appears that it has met its primary endpoint. For the rest of the talk, I will move on to my favorite discipline, immunology. Now, most of us here are Chinese origin, and Confucius once said, we all have two lives. The second one begins when we realize we only have one. This, for the young people, may seem to be a joke, but it's not. For people my age, and people like myself, and many of you, who have seen critical points in their time, either through their own health, their own career, or friends and relatives. But medical scientists also have two lives. The second one begins when we realize that immunology is the orchestra for most life symphonies. As is demonstrated that immunology is intricately involved beyond infection, autoimmune disease, transplantation, but now we know neurodegeneration and cancer. The fundamental point that immunology has to be involved from infection is that the T cell receptor is recognized as this little small peptide that could be part of a virus, a bacteria, a parasites. But more recently, we know that it could be part of a cancer cell. Of course, if the T cell receptor sees a part of your own cell peptide as being foreign and attack it, there comes autoimmune diseases and neurodegeneration. So I would say that the metabolic states and the DNA damaged state, which is a fundamental part of cancer, feeds back to the immune system because the immune system is so sensitive to any biological and physiological modulations that's happening. As it was said in the introduction by Professor Lowe, the laboratory stumbled across a gene called CTLA4 in 1995. Completely shocking. Everybody thought this is the gene that helps the T cell to be better. It turned out it was a break. You can see the mouse without the gene on your left have these pearls, jewels. They are T cells. The mouse dies in 2 weeks. Now, I am not a very smart person. I was cursing the darkness because in 1974, a professor by the name of Stutman at the Memorial Sloan Kettering said that mice without immune cells can fight cancer. And so immunologists forget that you can do anything. My friend, Jim Allison, lit the candle. He made an antibody. He blocked the break, CTLA4, and now the T cells go forward and not stop. That, of course, happened in many situations to go beyond what the T cells would do in an ordinary situation and start attacking the cancers. This, of course, led to tremendous success in many different cancers, but not in many others, something that we still have to do. And together, of course, with Tosco Honjo, who discovered PD-1, they went on to receive, very deservedly, the Nobel Prize as the fourth pillar of cancer treatment is now established. There are now, and has been for the last 10 years, 2,000 clinical trials to combine PD-1 with X and Y and Z, and combine CTLA-4 with A and B and C. Out of those 2,000 clinical trials, there has been a few successes, very few, and marginal. You can see here, by combining PD-L4 with PD-1, patients survive a little bit longer, a little bit better, but also there is increased toxicity. So I would say that as much as I would like to be optimistic, it has been very difficult. And overall, I would say it was a marginal success, and perhaps we could do better, can't we? Now, the tumor microenvironment is so complicated. Everybody is trying to say, I can do X, Y, and Z, and I can make the tumor microenvironment more responsive to the T cells killing the tumor more and longer. Why longer? Because T cells, like many of us, cannot work 24 hours a day, 365 days a year. We get tired. And so what happens when you get tired? When they get tired, they need an interleukin 7. Something, in fact, our laboratory discovered before the immune checkpoint, where people say, why would you even bother doing this? Immunology is not going to cure cancer. And just as a kind of a way of finishing off, a metabolism can activate DNA instability. The DNA instability is a hallmark of cancer development. But maybe we can even take advantage of this hallmark to our own advantage to treat cancers. First of all, let's go back to the isocitrate dehydrogenase, the metabolic enzyme. Our laboratory discovered that the metabolic enzyme can downregulate an enzyme called ATM, which is important for DNA repair. In fact, the co-first author is Wanda Lee, who is now the administrative person who oversees the Institute of Oncology Immunology across the street on 17W. Now, is that DNA instability causes cancer. So what we need to do is to repair the DNA. But no, we cannot repair the DNA. The DNA is broken, and the repair enzymes are gone. What are you going to do, Tak? Well, if they can repair this much DNA, why don't we let them repair that much DNA? And that way, they will die. But Tak, you're going to give my cancer patients more cancer. I'm sorry. They only have a limited time for which we can do something. And to prove this point, PARP inhibitors, discovered by Alan Ashworth, and also topoisomerase inhibitors are approved. So the idea that if they have problem with repairing DNA or taking the longer time to actually be able to finish the cancer cell's ability to divide, they will choke. So in this spirit, for the last 15, 17 years, we've been finding targets that are, number one, amplified in cancer cells, and number two, because that way you can get a therapeutic index, number two, they are fundamental to the cancer cells. Now look at these pictures. The top two are normal cells. They have 1 and 2 red pink dots. They are called centrioles. There's an enzyme there called PLK4. They are like two teachers who count the number of students. How many students are there in a normal class? 46, sorry. 47 for trisomies. But in the cancer cells, you can see the two bottom ones. How many students are there? 100. How many? Do you need more teachers? Yes. Do you need more ropes, which the teachers hook onto the students to count? Yes. These two enzymes, PLK4, TTK. So Carmen Wong, here in Hong Kong University, showed that in liver cancer, these enzymes are up 10 to 100-fold. So what are you trying to say? Make a drug against them. And indeed, for the last 10 years, we've been working with a lot of chemists, including Homer Pierce, who developed gemcitabine and Alimta in Eli Lilly before his retirement. These drugs against the two dots and the green lines are now in phase 2 clinical trials, and both of them are fast-tracked, and the company is based in Hong Kong. And we're hopeful that these preliminary data looking good is a RAS-mutated colon cancer at late stage, responding to the first drug. And here is breast cancer, recurrence late stage, recurring, and responding to the second drug. So can this genomic instability also activate the immune system? And yes, they can. Both of these drugs have been used by investigators in Hong Kong and at Harvard and other places to show they actually can activate the immune system. Let us go back to the very, very beginning, where it all started. Started with the T-cell receptor. The T-cell receptor, when they see the peptide at HLA, turn on all the go. No cells divide faster than T cells, 4 to 5 hours for a mammalian cell. They have 3 breaks. One break is HPK1, and Treadwell Therapeutics, based in Hong Kong, has now one in the clinic, finishing phase 1 clinical trial with some very interesting activities. So T cells and CAR T, they are kind of like Jason Bourne. They don't care about the tumor microenvironment. They go in and they find the bad guy and they kill them. CAR-T, 6 of them now have been approved, and patients 10 years ago, some of them are still alive. So CAR-T, unfortunately, has very few targets, CD19, BCMA. TCR-T, unlimited targets. But you have to find the peptide and the HLA. This is CAR-T. There is no question it works and been FDA approved 2017. Now, the targets for the TCR-T are a little bit more complicated. They could be mutations, but they could be viruses that cause cancer, or can be cancer testis antigens. GP100 has now been approved for uveal melanoma, a very, very bad cancer once it's spread. Been approved now for 1.5 years. NY-ESO-1 is looking very good, and so are the human papillomavirus in oropharyngeal carcinoma, etc., etc. So what can you do? What problem is there are very few T-cell receptors that can be found. And so we need to be able to enhance, and Treadwell Therapeutics has been able to do that in 10 years' time. And we can now basically see that we are fishing out like a fishing net, while others are fishing with a rod. And by looking at this, you can see that we have multiple TCRs that are really good because they go right down to the 0, and we can choose from dozens, while the black one is one that is pretty, is clinically active by a company. Now I'm going to end in my last 3 minutes that I would rather have questions that can be answered than answers that can't be questions. You can see here what is going on. The T cells are trying to get into the tumors, but there are actually walls that are blocking them. They are trapped in the blood. And what is the Trojan horse? The Trojan horse is the brain. 170 years ago, some Germans stimulated the nerves of dogs and decapitated humans. Excuse me, decapitated human. And the spleen moved. It turned out that there are these nerves that are sending messages to the spleen. And these nerves could make acetylcholine or norepinephrine. Together with a neurosurgeon in New York, Kevin Tracy, who's a genius, by the way, we worked together and show immune cells make neurotransmitter. In fact, after you infect a mouse with a virus, they make tons of neurotransmitter. But say, TAC, who cares if they make neurotransmitter? Do they really do anything? How do you prove they do something? Maureen Cox, a postdoc in the lab, deleted the gene acetylcholine transferase, which makes the neurotransmitter. Now this mouse has no neurotransmitter in the T cells. And now you give them a virus, they cannot clear it. Whereas if they have the neurotransmitter, they can clear it. How do they work? Many different ways. One of which is the T cells bring the acetylcholine directly to the endothelial cells and say, knock, 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 I have the key. Open the door, I'm getting out. Now this is going to be very beginning, I think, of a lot of things that's happening. Just very quickly, this is your liver. Your liver on the red are all nerves. Are the nerves doing anything? Oh no, who cares? And nobody cared for decades. Now if you look more clearly on your right, the nerves are wrapping around the portal veins. They don't care about the central veins. I think they're doing something. You may not agree. I will tell you. When we delete the neurotransmitter for the immune cells, the tumor in the liver grows faster and the mouse die earlier. Not only that, if you give the mouse high-fat diet, which is like, steak and other things, the tumor grows faster and it's dependent on the neurotransmitter made by the immune cells. This is human data. Liver cancer patients with neurotransmitter on their liver cancer cells live 500 days longer. My final slide, second last slide, is that I went downstairs to the cancer doctors and I said, can you measure the precursor of the neurotransmitter, choline plus acetyl-CoA makes acetylcholine. Can you measure it in your patients go under immunotherapy? And they say, why? I said, I don't know. Maybe the immune cells need neurotransmitter to fight the cancer. Don't be crazy, Tack. You are delusional. 2 years later, they measured it. And indeed, if you have lots of precursors to the neurotransmitter, you live, and if you don't, you die. So finally, I want to quote the African proverb. If you want to go fast, go alone. If you want to go far, go together. And these are the people. Thank you very much.